hopefully, yes. And I'll cheat a little bit. I'll have a timer because otherwise I'll just speak forever. So thank you, everyone. Thank you for being here. I'll just start the timer. And it's going to be very hard to keep up with Dongjun and Alyssa and Isabella because they just had fantastic presentations, but I'll try my best. So the work that I'm going to speak today is actually a combination of the last 4 years of work with my collaborators, especially Francisco Lobo from UFMG Brazil and now with NCI in the United States, but also our PhD student Lindbergh Leite at the University of Brasilia and Tel de Campos at Microsoft. And our work has been on a taxon-specific linear B-cell epitope prediction, and I'm going to walk you through what took us to get to phylogeny-aware transfer learning. So a bit of context that's probably, in retrospect, unnecessary given the audience, but just bear with me for my colleague data scientists in the audience who may not be as versed in immunology as the rest of you. B-cell epitopes, which are the focus of my work, are part of an antigen, like, for instance, a protein, which are the types of epitopes that I'm interested on, that binds to an antibody. Identifying epitopes is a crucial part of the development of many immunological products, downstream immunological products. And since the 80s, computational methods have been used for that. Originally, propensity score, physical chemicals features, but since the mid-2000s, with a pronounced shift towards machine learning, and in the last 5 to 8 years, as with everything else, deep learning. The focus of this work, the work that I've been developing with my collaborators, is on linear B-cell epitopes in the context of infectious diseases. So from now on, whenever I say epitope, please keep that in mind. I'm thinking about linear B-cell epitopes for infectious diseases. So I'm not looking at neoepitopes. I'm not looking at allergens. And most, if not all, of the widely used LBCE predictors to date have been trained and have been developed under a generalist assumption. Basically, they aggregate data from a wide variety of pathogens, and it is part of the design principles of those things. They get a lot of diversified data, and they aim to generalize to a lot of diversified pathogens. And actually, that is an explicit assumption in a lot of cases. There are quotes from not too old papers mentioning that to achieve bias-free prediction, you actually need data from a very broad range of evolutionary distinct pathogens. And I agree with that with an asterisk. So I asked my daughter to give me an image of polite disagreement, and she gave me the Nope Ninja. And I think that is true if you assume that your models are going to be deployed without any reference of the pathogen of origin of that antigen. If you assume that the models are blind to the pathogen, then yes, you need to train it with as diverse a data set as possible. But the pathogen of interest is always known at time of deployment. No one simply wakes up in the morning, grabs a random sequence, and tries to start predicting epitopes. It always happens in the context of a broader, more fundamental biological question that someone may be exploring. And not using that type of information in the development and deployment of the models, I think, is a waste of valuable information. So that informed our original hypothesis. And the hypothesis was that different pathogens would lead to different signatures in the feature space when it comes to which regions of any feature space would be more enriched with epitopes or non-epitopic peptides. And we did a quick exploration of that a few years ago. That was part of Jodie Ashford's PhD back in 2021 was the paper. And we grabbed a bunch of data on epitopes and confirmed non-epitopic peptides for different pathogens. We embedded those peptides into a feature space. We projected that feature space, that embedding, down to 2 dimensions for visualization. And once we had that, we stratified both in terms of positive and negative and by pathogen. And what emerged was very interesting because it resonated with our underlying hypothesis. Not only different pathogens exhibit different areas of high density of epitopes and non-epitopic peptides in the feature space, but also, and more critically, we do have overlaps between areas that are enriched with negative entries for one pathogen and positive entries for another. And if you don't distinguish the pathogens, if you just train the model on the combined data, what ends up happening is that those regions become confusing and the model is not able necessarily to classify properly entries that may lie in those regions. So that is something that we aimed to address. So like that gave us some encouragement, and we ran with that encouragement for the last 4 years, starting with very simple things, trying to build organism-specific, pathogen-specific models for a pathogen of interest. That works if you have sufficient data for that pathogen. So we did that for data-rich pathogens that worked beautifully, like very, very substantial, very solid gains in terms of predictive performance. Then we started exploring how little we could make the data. So how scarce can the data be and still give rise to useful models? And we found out that if you have more than a couple of tens of peptides, on average, positive and negatively labeled, you can still generate models that are competitive with generalist models that are trained on much larger volumes of data, but are not necessarily specialized for the pathogen of interest. And then last year, we published something that actually started a couple of years back to try and test two different hypotheses. The first one was whether or not our results were actually emerging from biases in the data rather than from the data generating process itself. So we generated predictors, validated them in silico, and then sent them down for in vitro validation with our colleagues down at UFMG in Brazil. And we sent them 9 targets, 8 of them came back as positive. So that gave us some encouragement that was better than our expectations, by the way. I would be happy with 2. I would have been happy with 2, but 8 out of 9 is not too bad. So that gave us some encouragement that the results that we were observing were actually part of the data generating process itself and not simply a bias in the data that's recorded on IEDB. The second thing that we tried to do with that paper, and we somewhat succeeded, was to get a pathogen that was really data scarce. So we focused on Mpox at the time of the 2022 outbreak, monkeypox, and we almost didn't have any entries for that. So we started aggregating data from phylogenetically related pathogens, from vaccinia, cowpox, etc. And we started going up the taxonomic levels and aggregating more data and seeing where the performance peaked. And at some point it peaked and then started going down again as we aggregated more data, so increased volume, but decreased pertinence to the problem. So that was quite encouraging, but it was still kind of an ad hoc manner of aggregating phylogenetically related pathogens. So that led a PhD student that I co-supervised, Lindeberg Faria from the University of Brasilia down in Brazil, to try and explore the idea of phylogeny-aware transfer learning. Basically, the core idea is quite simple, actually, but it worked beautifully, which is always a welcome thing. And we have a preprint currently, and the obligatory QR code is coming later in the presentation, so don't worry about that. It's in the second round of revisions now. But the basic idea is quite simple. So essentially, we originally given a target pathogen or a target group of pathogens, a target genus or a target family, we isolate every data point that we can get from IEDB that belongs both to the target and to a higher level taxonomic group that contains that target pathogen. Then we separate those things. So we have higher level sans or without the target, and then the target pathogen. We use the data from the related pathogens to fine-tune the ESM2 transformer, the ESM2 embedder, to the task of epitope prediction, linear B-cell epitope prediction, but with a bias towards that particular taxonomic group. Then we deploy that fine-tuned ESM embedder to calculate features for the target group, and we train a pathogen or group-specific model based on that. It's quite simple. It's not rocket science, but it works, which is always a welcome thing. And I'll show you why it works, or not why, but how. So we got 19 dataset pairs, so target group and a higher level taxonomic grouping, including viral, 6 viral, 8 bacterial, and 4 eukaryotic pathogens. And for each of those, we fine-tune the ESM embedder and develop a pathogen or group-specific predictor. We use similarity-aware data splitting, so based on homology to prevent homology-driven data leakage. And the splitting was quite early to prevent data leakage through pre-processing or modeling as well. Cross-validation, homology-aware cross-validation for model development and optimization, and a test set, final performance on a test set that was isolated quite early into the process. We fit the factorial ANOVA. The assumptions of ANOVA miraculously were all satisfied, so everything, the residuals are normal, identically distributed, homoscedastic, so the dream, textbook dream of data, fortunately. testing for differences on methods, differences on ISM version, because we also wanted to test that, and using dataset as a blocking variable to remove the between datasets variability from our analysis. And we also done it post hoc comparisons, all versus one. And we tested against two types of baselines. The internal baselines that we developed were to test specific questions in terms of why our models were working, if they were actually working. So we developed two baselines. The first one is we called ESM baseline, which is basically the same idea, but without the ESM fine-tuning. So we're using the raw ESM features and then just building the group-specific predictors downstream, but without fine-tuning. And that was, of course, to test the effect of fine-tuning. The second internal baseline, we called it the non-phylogenetic transfer baseline, NP transfer. And it's the same modeling approach, but fine-tuning the ESM embedder to the epitope prediction task, but breaking the phylogenetic link. So essentially fine-tuning to epitope prediction, but using unrelated. So if we're trying to develop a model for a virus, we fine-tune on bacterial and eukaryotic pathogens. And that's to test the effect of the phylogenetic assumption there. And we found effects. We found a systematic gain, in both cases statistically significant, with an effect size in terms of absolute gains of about 0.03 against the non-phylogenetic transfer, 0.33 for the ESM baseline. When you look at the normalized effect sizes, it's about 0.7, so like 0.7 standard deviations for ESM baseline, 0.5-ish standard deviations for NP transfer. So that's interesting because that gives us some sense that what we're doing actually carries some predictive value. And we then compare that against the current state of the art in linear B-cell epitope prediction. That is BPyPred 3.0, epitope, epitope 1D, and epitope VEC. Some of those are deep learning based. Some of those are engineered feature, kind of the domain-specific feature based. And we ran this comparison using two comparison data sets. One is the same test sets, the paired test set, which is the standard practice. But we noticed that our test set, which the epitope transfer, the proposed approach, did not see during model development, contained entries that were part of the training set of the state of the art. So the assessment on that for the state of the art is biased upwards in terms of performance. So we extracted a second test set, which is the non-leakage test set, which essentially for each of the state of the art does not contain any entries that were part of their training set. And the effects here are substantially more pronounced. So we have differences in terms of AUC ranging from 0.09 to 0.13 absolute values, and in terms of normalized effect sizes all the way up to like about 1 standard deviation, 1.1 standard deviation. So quite large effect sizes there, evidencing the kind of the difference that this sort of training regimen provides. Just focusing a little bit on the paired differences of performance. So this is already removing the between data set variability. We have substantial gains, and in a lot of cases, substantial gains that are uniform against every other state-of-the-art model. In some cases, we have gains against one and modest losses against others. We set a plus or minus 0.05 AUC as a region of practical equivalence for that comparison. So we are not focusing any gains or losses within that gray band there. We are not terribly worried about them, because like plus or minus 0.03 or 0.05 is kind of within the margin that you would, okay, like I can use this or can use that. But we had a lot of cases in which we had systematically high gains in performance for the proposed approach, epitope transfer, in relation to the state of the art, especially for filoviruses, which was quite surprising. Filoviruses is a family that contains Ebola and Marburg, so like, reasonably relevant. And we observed like ridiculously large gains. It's consistent with random distribution of effects. So we tested that. We modeled the gains as a random distribution, and having kind of high gains and some losses is consistent with what we would expect there. But there is a systematic bias towards gains in predictive performance. Just to wrap up, the conclusions from this latest iteration of our work, like training pathogen or taxon-specific predictors improves performance, which shouldn't really be surprising. The interesting bit there is that we can do that with a small fraction of the total data volume. that it takes to train a generalist model. And it becomes a specialist model for a group or a pathogen of interest. We also managed to conclude that ESM2 fine-tuning does give a predictive edge, and it's even better if it's paired with a taxonomy-aware data filtering strategy before the fine-tuning. And finally, it does result in substantial gains for at least some pathogens. Majority of pathogens in this specific case, but the interesting bit is that for any pathogen that you're trying to develop these models for, you can always run a comparison. So you can always know if that model is expected to give you better results than a generalist or not, which would guide the choice of the most appropriate tool for any downstream question that you're trying to explore. That's basically it, and I managed to keep the time, which is brilliant. So thank you very much. I'd like to just extend my thanks to my co-authors there, Lindeberg, Theo, and Francisco. As I said, obligatory QR code. So if you want to check the preprint, there's a link to the source code and the notebooks in there as well. So thank you very much.
Thank you very much, Felipe, for a great talk. Questions?
Hi, thank you very much. Very interesting talk. I was wondering if I'm not sure if you've tried this, but if you could also see gains in specialized epitope prediction, if not doing it by taxon, but doing it by like the type of antigen. So like the type of protein that it was, like the structure of the protein, whether or not there's like a similar sort of thing where like when you have a specialized epitope prediction model, it gains performance.
Yes, there is. So I had a PhD student that was supposed to explore that question a few years ago, but he essentially went on to do better things. But yes, some of the state-of-the-art models that I compare, at least one, epitope 1D, those use antigen, those use protein similarity as a first line of filtering. So there is value there, and those things are complementary. So like a first approach would always be to try and see if there is already an epitope that's validated on a highly similar sequence. So incorporating that into the pipeline would increase the predictive signal, but testing that without data leakage is kind of complicated testing for the performance of the models as well. But yeah, I do see value there, but we did not incorporate that into our pipeline, mainly because testing is a nightmare. We wouldn't be able to know if the signal that we're getting is because we are actually predicting something or simply because of homology-driven leakage.
Yeah, so I have a question related to a contradiction you mentioned at the beginning, so that the identical or very similar linear epitopes would be different, can be positive or negative in different pathogen contexts.
Yeah.
And I was wondering, what is the biological explanation for it? Is it like...
That's an excellent question. That's an excellent question for which I do not have an answer. But I should probably preface that I'm not a biologist. Like I'm a data scientist. But like that's something that has, like in the very first paper, one of the reviewers asked precisely that question. So what's the biological rationale? So one of the things that I should clear is that it's not that very similar peptides will be positive in one context and negative in another, but peptides that map to similar regions in a feature space, which may or may not be similar in the space of sequences. It depends on that mapping between sequence and features. But it may be because those patterns are cryptic. It may be because those patterns occur in proteins that are part of different parts of the life cycle of the pathogen that are more or less exposed to the immune system environment. But to be absolutely honest, I don't have a good answer for that. Like we know that there is a predictive signal there, but the causality of that remains unclear, at least to me. Maybe someone in the audience has better insight, but I don't.
Thank you.
No problem. Thank you.
Felipe, I have my own question.
Yes.
Is any of this framework generalizable or extendable to T cells?
Possibly. Like T cells, they go through the whole processing part, and they are at the same time easier and more difficult to work with, more the whole bit about the differences in prevalence in different populations, the haplotypes, but easier because they are more constrained in terms of their length, in terms of certain properties, in terms of the key residues that make up for a T cell. But my impression is that the phylogenetic contribution, so the contribution of incorporating phylogeny into T cell epitopes, is likely to be less pronounced than it is in B cell epitopes because of the whole processing that goes on. I was having a conversation with someone over lunch earlier today from Norway, and she was asking about that. And my impression is that the more, potentially the most relevant thing is expression, like the expression pattern of certain proteins for T cells, much more than phylogeny. But I'm not totally sure about that.
Yeah, I think that's very interesting. More questions? Well, then with that, we thank Felipe again.
Oh, thank you. Thank you for having me.